Hello friends, welcome to my channel Dr. Sam's Biology. In this video, we will discuss how this microRNA was discovered. We know that this discovery got Nobel Prize in 2024 in the category of physiology and medicine. So first, what is microRNA? These are small RNA molecules of 21 to 23 nucleotides. And these are non-coding, means they do not form any type of protein. These are non-coding RNA molecules which are small. They are 21 to 23 nucleotide in their length, and they are found in plants and also animals. They are involved in post-transcriptional regulation, means they regulate the expression of gene after its transcription. So these are our microRNA molecules. Now coming to how they were discovered. These two scientists, Victor Ambrose and Gary Ruth, they were working on a tiny board that is C elegans. So they were working on this C elegans. We know that it is a model organism in biological studies. So they were studying this only. Both these scientists found that this tiny board, that is C elegans, uses these two genes, lin4 and lin14. It uses these two genes to progress from larval stage 1 to larval stage 2. So for its progression from larval stage 1 to 2, it uses two genes, lin4 gene and lin14 gene. If this lin4 gene is inactivated, if it is mutated, then this larva stuck in L1 stage. If this gene is defective, then this larva cannot progress from L1 to L2. But if this L14 is defective and this one is normal, just opposite to this, if this is inactive, then it will enter into the L2 stage prematurely. So it will enter prematurely. Before its maturation, it will enter to L2 stage. So these both genes, lin4 and lin14, these genes have opposite function. Lin4 gene needed to leave L1 stage and move to L2 stage. So this gene is needed to reach to L2 stage. And this lin14 is needed to complete L1. So this is needed to complete this stage, and this one is needed to reach this L2 stage. So both these genes have opposite function. It is quite normal in biology. Many genes have opposite function. When they found this, both these scientists thought that it is a protein. They thought that this lin4 and this lin14, they are forming protein, and this protein is doing this function. But the first surprise came when they found this, that this lin4 gene is actually a forming RNA. That is 22 nucleotide RNA. This gene, which gene? lin4 gene, is forming a RNA. And this RNA is not forming any type of protein. And this 22 nucleotide RNA actually binds to the mRNA of this lin14 gene. So suppose this is lin14 gene. If this lin4 gene is inactive, this is not present, it is not expressing, then it is expressing, it is forming mRNA and forming protein. And this protein is doing its function. But if this is active, then is it forming a 22 nucleotide RNA, and this RNA binds to this mRNA of lin14 gene and makes this inactive. So if it becomes inactive, then it will reach to L1 to L2 stage. So first they found this. They were the first scientists that found this microRNA that acts as a ribose switch that activate or deactivate this gene by binding it with its mRNA. So first they thought that it is unique to this tiny one, this C elegans, but after some time they found that it is present in almost all the organism, and it is of great utility to humans because it can be used in many diseases such as cancer, HIV, many genetic diseases. You can say that any mRNA can be silenced because anything that is defective, it expressed through a defective protein or defective RNA, it can be silenced. So this is also called gene silencing. So for this discovery, Victor Ambrose and Gary Rupun, they got Nobel Prize of 2024 in category of physiology and medicine.